going to be in 3 parts. So first we're going to talk about what is a coronavirus and how does it spread. And then we're going to talk about some public health measures that we're taking to slow down and hopefully stop the spread of the virus. And then last we're going to talk about some potential therapeutic interventions that people have been working on. So let's start with the basics. What is a coronavirus? So it's a virus that's pretty large as viruses go, and it has an RNA genome with a positive sense. And that means that that RNA genome basically acts like an mRNA when it gets into a human cell. So the ribosomes can just grab onto that RNA genome and go to town and start translation. It's enclosed in a membrane, which is going to be really important for how it spreads and also what we can do about controlling it. And the corona part means crown. That's made up of glycoprotein spikes. So there are a bunch of spikes that are sticking out of the virus membrane envelope, and that's what's going to interact with the cells. And so this is a schematic of it, and then we have an artist's rendering of what this thing looks like. Here's a schematic of a coronavirus genome. And this one is not for the current one that's going around. This is for the original SARS virus that caused an outbreak in 2003. But it is very similar. Some details are different, and if you're interested in the sequencing work that's going on, I encourage you to follow Trevor Bedford's Twitter feed. So he's at the Hutch in Seattle, and he's been doing a lot of the work on sequencing the viruses that are going around in Seattle and tracing its evolution. But the basics of how this works is there are 2 overlapping open reading frames that code for the non-structural proteins. So notice they are overlapping. So you're going to have ribosomes reading through to here, and then you're going to have ribosomes starting at this point and reading to there. And then there are a bunch of other structural proteins that are made by the other part of this genome. And the 4 structural proteins are the spike protein that we already talked about. There's an envelope protein, membrane protein, and the nucleocapsid. And so it's pretty simple. This thing doesn't have very many proteins. That's pretty typical of a virus. Mostly it gets your own cells to replicate it, and it uses all of your cellular machinery. Yes?
When you say overlapping open reading frames, does that mean they're in tandem on the same sequence, or are they actually?
They are on the same sequence. So to read this one, the ribosome is going to start here and stop there, and then to read the second one, it's going to start back here, which is still inside the first one. Make sense? So, viruses are parsimonious. They don't have a lot of genetic material to work with, so things like that are pretty common. How did this pandemic start? So as is typical for a lot of new viruses that go around, it started from another animal and then went through some mutations that enabled it to be transmitted to humans. And right now, people think that the possible origin are pangolins. It's also pretty similar to some bat viruses. And I put a paper about that in the Canvas site, so you can check it out if you're interested. So it's also very similar to the first SARS coronavirus, and also MERS, which caused an outbreak a few years ago. And I want to point out that the flu virus is a different type of virus altogether. So people have been comparing this virus to the flu in terms of infectivity and how dangerous it is and things like that. But we shouldn't get confused and think that it's the same kind of virus as the flu, because it's not. And so that means that flu shots will not protect against it. It's still a good idea to get a flu shot because you don't want to get sick and have to go to the hospital when other people might need the resources. But it is not the same kind of virus. Question?
Also, could flu or any, basically getting the flu weakening your immune system and make it worse?
Yeah, that's possible. Certainly it's not a good time to have any kind of sickness. Let's talk about how it actually spreads. So the virus is most concentrated in the upper respiratory tract, and the virus can survive on hard surfaces for up to 2 or 3 days. It survives a lot less time on soft things like paper or cloth or something like that, but it can last on hard surfaces for quite a while. So this is a good reason to wash your hands before eating, after you've been touching any kinds of doorknobs or countertops or anything like that, and disinfect everything pretty frequently. It can also spread from person to person through droplets, and that can be either a cough or a sneeze, and also, from touching an infected person. The main route to getting it is touching your nose or mouth. So, yes, you can breathe it in through aerosol droplets, but a more likely form of transmission is touching surfaces that are infected or another person who has it and touching your face. So it's important to disinfect stuff, wash your hands, try not to touch your face in order to slow the spread. Yes?
What is the mechanism that explains the difference, the large difference you said between a survival time on a hard surface and a soft surface?
So it has to do with that envelope, the membrane on the virus. So if it's on a hard surface, that membrane can stay relatively intact and keep the virus together. Whereas if it's in something porous like paper, it's gonna have a lot of intermolecular interactions with the fibers in the cloth or the paper, and so it doesn't last as long. And I want to point out that this information is from a preprint on medRxiv, so it's not yet peer reviewed. I checked it out for you guys, it looks good, but you should be careful with stuff that's in archive because it's brand new and it's not peer reviewed. So you can get some good information there, but you have to use your judgment about it. So here's their data from this paper. So they looked at how long different viruses can survive in aerosols. And so this is the new coronavirus. This is the original SARS virus. So this one actually is a little bit less stable as an aerosol. That's good news. But it looks like it might be a little bit more stable on various hard surfaces. So this is virus titer, so that's the amount of active virus. And they tested that by actually putting it into human cells that are cultured and testing how many got infected. So that's a real measure of active virus. And then this is the hours post-application for different kinds of hard surfaces. So you can see for cardboard, it's kind of all gone after 48 hours. But for things like plastic and stainless steel, there's actually a significant amount after 2 or 3 days. So that's why it's really important to wash your hands, disinfect everything, try not to touch your mouth and nose. Let's talk about how it gets into cells. So it is now known that the entry to cells goes through a protein called ACE2, and so that's referred to as the receptor for the virus. Of course, as far as your body is concerned, that is not a receptor for the virus. It's a perfectly innocent membrane protein that has its own biological function, and the coronavirus is hijacking it in order to get into the cell so that it can be replicated. So what ACE2 actually does is it's called angiotensin converting enzyme 2, and it's a membrane-bound protease. And the function of that protease is to cut up the peptide hormone angiotensin. And that hormone is involved in the regulation of blood pressure and also vasoconstriction. So it's one of these things that's expressed as a prosequence, and then it needs a protease to cut it up into the mature form. And that's what ACE2 is doing. And it's on the outside of cells, which makes sense because it's processing a hormone that's going to go on to do some signaling. So before this coronavirus outbreak, the actual structure of ACE2 was not known. Nobody had solved it. But recently, a cryo-EM structure was solved. And this is the structure of the ACE2 protein, and it's also bound to this neutral amino acid transporter. And the neutral amino acid transporter is not involved in coronavirus infection. It's just a protein that binds to the ACE2 and keeps it stabilized. And so it was necessary to include it in the construct for the cryo-EM structure in order to get a reasonable structure. So here's the actual ACE2 protein. It's a dimer. It's a homodimer. The 2 monomers are identical. They're shown in different shades of blue here. And that's the ACE2 protein on top. PD is the protease domain, and CLD is this membrane anchor down here. So notice it has, this is the part that's actually going to do the cleaving the hormones. and then it just has this little helix sticking down into the membrane, and then it also interacts with the neutral amino acid transporter that has a really big transmembrane part. So, again, this part is just there to make solving the structure easy. This is the part that we're actually interested in. So that's just the ACE2 protein by itself. Here's how that interacts with the coronavirus. And again, this has no biological function that we want. This is just the protein that the virus evolved to hijack in order to get into cells. So the coronavirus has a receptor binding domain, that's in orange here, and it binds to the top of that protease that's sticking out into the extracellular space. And these proteins are heavily glycosylated, and that's probably important in some of these interactions. If you're interested in that, you can go look at the paper. I posted it for you, and you can see details of what the intermolecular interactions are. But for now, I just showed them in sticks. And it's also important to remember that the virus doesn't enter through this little neutral amino acid transporter. This thing is little, and it's got the virus, like we're just showing a tiny little receptor binding domain. It has a whole huge thing attached to it. So it does not get in through this channel. What it does instead is it has to fuse its membrane with your cell membrane. And that's a pretty complicated process that has a lot of steps. But this is the first step. So that's the first. So here's the most important one. Wash your hands a whole lot. So it's important to use soap and really thoroughly scrub your hands. Remember that membrane envelope of the coronavirus. We have to use soap and really scrub it off in order to break down that membrane and inactivate it, and then also wash it off and wash it down the drain. And so it's important to wash all the surfaces of your hands really thoroughly and scrub for at least 20 seconds. And there are all these helpful memes going around with songs that you can sing for 20 seconds to make sure that you do it long enough. And I think that's really helpful. If you aren't able to wash your hands, hand sanitizer or wipes are a good second choice. They need to have at least 60% alcohol in order to break down that membrane of the coronavirus. It's not as good as actually scrubbing and rinsing, but it's better than nothing. And if you're in a situation where you can't wash your hands, that's good. And then again, try not to touch your face. It's really hard. Humans touch their faces all the time. And I've been trying to not do it for the past week or so, and it's really difficult to break the habit. The reason this is so important is that those ACE2 proteins that I mentioned, we only have those in our respiratory system. So no other part of the body has the receptors to actually let that virus enter. So if you just get it on your hands, it can't do anything. And washing it off is completely going to take care of the problem. So it's just really important to make sure that it doesn't get into your nose and mouth. So I mentioned hand sanitizers and wipes are a good plan B. They do need to have at least 60% alcohol. There are various alcohol options that go into them. How it works is by breaking down that lipid envelope for viruses, and of course they also damage bacteria. There are other kinds of hand sanitizers that don't have 60% alcohol. They have other active ingredients that are antibacterial. That doesn't help so much with the coronavirus. You really need that alcohol to break down the membrane and help wash it away and inactivate the virus. I should also point out just for future reference that these things are less effective on other kinds of viruses that don't have a membrane coating. If you just have a protein capsid, then alcohol is not as damaging to that. So let's talk about why we canceled finals and everything is kind of shut down for the time being. In order to understand that, we need to understand exponential growth. And exponential growth is really counterintuitive for humans. We're very good at thinking about things that grow linearly. And things that grow exponentially are really hard for us to picture. But so in order to picture it, we're going to talk about a good old example of a lily pond that has some lily pads in it. And so imagine a situation where you start on day one with one lily pad. And then the second day, there are 2 lily pads, and the population doubles every day. And we know that the entire pond will be covered in 30 days. So on what day do you have half the pond covered? Day 29, obviously, right? But so let's think about how weird and counterintuitive that looks when we look at the actual exponential growth curve. Okay, so we know that half the pond is covered on day 29, but if you look at, say, day 23, so we're a week out from having the entire pond covered with lily pads. And at a week out, it's way less than 1% covered. So you would not think that the lily pads at that point were going to cover the pond or even cause any kind of a problem. And also, depending on where you're standing, even if you looked at the pond every day, you might not even see a lily pad. And that's a week out from this. So we have to be really careful with things that are in exponential growth mode. And that means that precautions that we take can seem, really overblown given what's currently going on around us. But we have to make sure that we don't get to here. So let's look at that with some examples from the coronavirus. So this is a plot of confirmed cases from different countries. I got this data from NPR. It's a couple days old, so things have changed since then. So we do have exponential growth in several countries. So in Iran and Italy, the number of cases are really spiking exponentially. Japan and Singapore instituted measures to keep people distant from each other and slowed down the growth sooner. Early response in the US was a lot more like what happened in Italy. And we have a low number of cases right now, but some of that is due to not having enough testing. And also, again, we're in the early phases of the growth, so it's difficult to see exactly what's going to happen. This is the same kind of a plot, but with confirmed deaths from the virus. And this plot is from Sean Ryder at UMass Worcester. So if we look at the confirmed deaths, there are some really scary numbers going on in China, which has finally started to level off, and Italy. South Korea appears to be growing much more slowly. And in the US, our outbreak started later, and so we're still not sure exactly what's going to happen. And I put this up, not to scare everybody, but just to remind everyone that this virus is very dangerous. It's not just something that is going to give you cold symptoms and be unpleasant for a while. Actually, for everybody in this room, it probably would, because young people or even middle-aged people are not tremendously vulnerable to dying from the disease. It's older people who are at the most danger, and so it's important not to spread it in order to protect older people in our community. Okay, so how does social distancing help? Question in the back.
Real quick, I heard even if you get it and you recover, your respiratory will be scarred or damaged, so it won't be the same. How long you're predicted to live will decrease, right?
That's a possibility. So that doesn't. The question is, there could be scarring or other long-term effects to your respiratory system. That doesn't happen to everyone who gets it, but it can happen. So it is a risk. So yeah, even if you don't die from the disease, it could cause some really bad long-term health problems. And so, so we don't want that. So yes, everything is kind of shut down. We're at, we have social distancing measures here at UCI, even though we don't have any confirmed cases. It's just being safe. Yes?
Is there some threshold that, I guess, around the world they not maybe cumulatively agreed on, but just chose? Because I know that we had these SARS earlier, in the 2000s, but I guess my question is is there a certain threshold that they pick to say okay, well, we need to start these measures because it didn't happen earlier? So.
That's a really good question. So, so we didn't have kind of a worldwide implementation of social distancing for SARS. And a big reason for that is that they managed to contain the spread sooner. So places that did have a lot of cases absolutely did take these measures. And that's why some of these other countries like Singapore and South Korea and Japan had such a good response to this outbreak this time, because they went through SARS and they had serious outbreaks and they learned how to do it. And so that's one reason that they were able to spring into action and take these measures. So here's what we're trying to do by implementing social distancing. So no matter what, people are going to get it. We have community transmission in the US. But the thing that we can avoid is having everybody get it all at once and overwhelm the hospitals. And that is unfortunately what's happening in some parts of Italy right now. There are people who are not getting the hospital care that they need because there are just only so many ventilators and so many hospital beds. And so if we take steps to limit the transmission, even if a lot of people get sick, at least we can hope to spread it out over a long time so that everyone can get the health care they need. And so that's why we're doing this. Okay, so those are kind of the public health measures that that we can take to stop the spread temporarily. And
do phage display using the ACE2 as code for the phage to bind to the cell quicker to organize the protein, the viral protein.
So you think it would be good to make an assay where you have the phage display of the ACE receptor and then pull out the virus?
So you can recognize the viral membrane that become part of the cell.
So that's a good idea, but it would take too long for this because PCR is really fast. Where if you had to make a phage display system that displays that ACE2 protein, it would probably take a long time. It's hard to phage display membrane proteins. It's a good idea, and you could do it. So that's something. So the Weiss lab has worked on phage displaying membrane proteins, but it's a project. It takes a long time to get it to work. So yes, you could do it, but it wouldn't be fast enough for the situation we're in now. Yes.
Do you know the structural difference between them yet, like completely, between like SARS and this virus?
So that's a really good question. We know some of them, and actually I'm going to show you a paper that was published yesterday. And there's a whole bunch of stuff in the PDB that hasn't been published yet, but the structures have been released, and you can go look at them. But yeah, I'm going to show you some of the things that are known. Some other question?
It was similar.
Okay, cool. All right, so let's talk about some potential therapeutic interventions. So all the things that I've been talking about, social distancing, being really careful about disinfecting everything, that's what there is to do when we don't have a direct ability to actually fight the virus. And we have to do it. It's the best thing we have right now. But it's also really important to come up with long-term measures that are actually going to control this disease. So what are some things that you can do in order to block a virus or keep it from being active? So you can bind it with an antibody. You can just block it from getting into the cells by inhibiting its interaction with the receptor. You can inhibit a critical protein that the virus needs, and it doesn't have very many, so there's a limited number of targets that you can start with. The very best thing is to develop a vaccine, and lots of labs around the world are working on that. That's going to take some time though, so this is not going to be something that happens right away. A better option is to screen a whole bunch of existing antiviral drugs, or even drugs for other things that are already FDA approved, and see if they can do anything against this virus, and that's what a bunch of people have been doing. Okay, so let's talk about some of the structural differences between this virus and other coronaviruses that have caused outbreaks in the past. So this is a cryo-EM structure of the spike protein. It was published yesterday, so this is very new work. The spike protein is a homotrimer, and the chains are identical, but they're not all in the same orientation in the complex. So in two of the monomers, they have that receptor binding domain pointing down, and in one, it's pointing up. It's ready to bind the ACE2 protein. And so there it is right there. So we saw that before in the context of looking at the ACE2 protein structure. And so then, again, this gives you an idea of how big the virus is compared to that protein. So we saw this little receptor binding domain on top of the membrane protein, but then you have to remember that that's attached to this whole complex, and that is just one little spike that's sticking out of the envelope of the coronavirus. So again, the virus can't enter your cells through that transmembrane protein. That's just how it gets a foothold, and then it has to have membrane fusion in order to actually get in. But so here's what this spike protein complex looks like. And here's a schematic of the different parts of the protein, and the ones that are in white were not included in their construct. And then there's some other stuff that's in white in this model where they couldn't quite get they couldn't quite see the protein. So the colored parts are what they have a good structure for. And so now to get to your question about what's different about it from other viruses, if we look at details of different parts of the spike protein, the overall folds are the same, but there are some interesting differences in local conformation. So for example, if we look at the receptor binding domain, here the green one is from the new coronavirus, and the one that's in white is from the previously observed SARS virus. So they have kind of the same shape overall, but there's a lot of conformational differences in these loops over here, and that might be really important in terms of how it binds. Same thing for the N-terminal domain. Same overall fold, but there are some differences. And you can look at that for other different parts of this complex. I also posted this paper for you if you want to read it. And so the question is this important for function? So we talked before about comparing protein structures and trying to figure out, which structural differences are important and which ones are not. It's kind of hard to tell just by looking at it. You have to do some experiments. And so in the same paper, these researchers measured binding of the receptor binding domain of this protein to the ACE2 receptor. And so you can see, we don't have time to get into what this assay is, but you can see that there's binding to the ACE2 receptor. And then if we look at the structure of the receptor binding domain, in this case, we're looking at the structure of the 2003 SARS virus, and the residues that are labeled in red are ones that are different in the new virus. And you can see that there are a lot of differences. And so that suggests that there might be some differences in how it binds to the receptor. And unfortunately, that turns out to be true. So this is the same assay as was done here. So this is basically a positive control. They're looking at binding to the ACE2 receptor, and we know it binds that. But then down here, we're looking at binding to the new coronavirus by antibodies for the 2003 coronavirus. And unfortunately, we can see that they don't bind at all. So new antibodies are going to be needed in order to control this virus. Yes?
Is it faster to develop the antibody than the vaccine itself?
So developing the antibody is an important part of developing a vaccine. And then also, at this point, everybody should just try everything in parallel. It's going to be important to have a lot of different approaches, and then hopefully something will work.
Wouldn't developing the effective antibody that binds to the vaccine and making a spray for the respiratory channel be an easier way to reduce the number of vaccines, the number of virus that was spread?
So developing a spray for the respiratory tract is actually really difficult. I mean, that's something that would happen at a much later phase of the drug development. To get a vaccine is going to be, it might take half a year or even longer. And, the early stages of that are probably not going to be in the form where you can just use it as a spray. We mostly don't even have that for seasonal flu. That's much more difficult to develop. Yes?
You said it binds to ACE2. What exactly does ACE2 do? Could we get rid of it and still be fine?
No, we could not get rid of ACE2. So its biological function is that it controls the blood pressure. So it cuts up that hormone that regulates your blood pressure, and it controls vasoconstriction. So no, we really need that thing. It's the virus picked a good target in terms of something that's very important to the cell. Yeah.
My question is going to be, so is it more, or I guess what happens if they continue to check all of the previous antibodies that we've used and we exhaust all those and we haven't found anything successful? Then would they turn to developing new antibodies, or?
Yes, so if the previously developed antibodies aren't working, we definitely need to get new ones. And the good news is that people are already working on that. So this is all happening in parallel. I mean, every lab that works on viruses has kind of dumped everything and started working on this, which is great. And you know, there's actually, if you look at Twitter, there's a lot of really interesting stuff going on about the latest work with this, and you know, people are really working together and sharing their results. So it's you know, it's really kind of inspiring to see everybody working together to solve this problem.
So then, is there any harm of so many labs trying to test the coronavirus? Like, are they actually ordering these viruses in containers to test them, or are they doing it a different way?
So how you get the samples? Okay, so the labs that are doing this are very well prepared to handle biological samples like that. So there are different levels of biosafety, you know, that you need in order to handle different kinds of biological agents. And my lab is BSL-1, which means that we can handle lab strains of E. coli and things like that are not particularly infectious. In order to do this stuff, you need to be at least BSL-2. And so there are a lot of procedures for safe handling of the samples. And you know, the people who are working on this already had that. They were already working on other viruses, and they know how to get samples and handle them safely and not spread it around further. Yeah, you're right. If people who don't have experience in handling human disease samples suddenly started doing this, that could make it worse. But nobody's doing that. It's against the rules, and I don't think anyone would do it. However, you can make the proteins and work on that. That's you know, that's completely fine. You know, just one protein by itself is not going to cause any problems. So here's one more therapeutic path that I want to talk to you about a little bit. This is another one that's from a preprint. It's not yet peer-reviewed. And what's going on here is this is an X-ray crystal structure of the main protease from the coronavirus. So I mentioned that we have those, you know, that the coronavirus has these different open reading frames that make long polypeptides. And that means that a lot of the proteins that it needs are expressed in all one long string. And then the main protease has to come along and chop it up into the individual proteins after that's done. And without that protease, you just get a giant polypeptide string that's kind of useless, and it'll get sent to the lysosome and degraded. And so that's another way to inhibit viruses. That's a strategy that was pretty successful in developing some of the first HIV drugs. A lot of the first HIV drugs that started having an impact are protease inhibitors. Yes?
But aren't those our proteases, so they would affect other functions as well?
So that's a really good question. So some of the tools that the virus uses belong to our cells, and so yes, it would cause all sorts of problems. These proteases belong to the virus. That's one of the few proteins that it has that's encoded in its RNA. So, you know, if you can block that, it doesn't harm us, and it does shut down the virus. So this particular main protease is a cysteine protease. It's a homodimer, and this lab solved a crystal structure of it, and they also used molecular modeling to design an inhibitor, which they called N3. And here it is bound to the active site of the protein. And so, since we spent a lot of time talking about cysteine proteases and how to inhibit them, you definitely have all the tools you need to go read this paper and download all the structures in the PDB and look at it and understand it. And I encourage you to do that. It's pretty interesting. Again, this paper is not out yet, but I checked it out and posted it for you. So in addition to their designed inhibitor that they did using molecular modeling, this group also screened a whole bunch of other drugs that were already available for different things to see whether they work against the main protease. And it turns out that some of them do. And this drug, Epsilon, works even better than their designed inhibitor. And so, you know, here we're looking at dose response curves for how much that inhibitor blocks the action of the protease. And so we can see that the Epsilon is, you know, it's almost a factor of 2 better than the one that they designed. So there are some possibilities, and you know, people are making progress on doing something to stop the virus. And so you know, that's another reason why social distancing is important and slowing down the transmission, because not only can we prevent everybody from going to the hospital at the same time, but the longer it takes for the disease to spread, the longer you can buy time for the researchers to come up with things that will actually treat it rather than just slowing it down. Okay, that's it for today. Have a great weekend, everybody. Have a great trip back to wherever you're going, and stay safe.